the female part of the flower. That is what is taking place inside the female part. So in the female part, the two main process takes place. That means the formation of gametes, which is known as the megagametogenesis, which takes place inside a female gametophyte, and megasporangium or megasporogenesis, and how the megasporogenesis takes place. This things is very commonly takes place inside an angiosperm. So when we are coming to this female part of the flower, two three things you have to know. First of all, as per the examination point of view, it is very important to go through the diagrams, what is given inside these diagrams. So if you see here, you can see some, excuse me, I'm using this PDF file so that I can edit something. Yeah. So yeah, I hope, yeah, it's correct. Yeah. So you have to see this female part of the flower. So if you see this pictures, diagrams, you can see something. They have given a simple female part which consists, which we commonly called as carpels, and if there is only one pistil, we call it as monopistillate, and if there are more pistils, we will call it as multicarpellary. Then if the pistils are fused, we will call it as syncarpous, and if the pistils are free, we will call it as apocarpus. And if you see here, you can see that this is having stigma. This is a female part of flower. Stigma is there, style is there, ovary is there, thalamus is there. Stigma is the area where the pollen grains come and fall. Ovary is the area where the female gametogenesis takes place, which is having megagametogenesis, megasporogenesis. Those takes place inside this ovary. And inside this ovary, we are having ovules, and these ovules are attached to the plant through placenta. You have studied that in first year. And this this ovule is having a structure. This is the structure of an ovule, and this ovule is anatropous. Anatropous means which is inverted. That is a word meaning of this particular thing. And if you see here, this particular ovule is having different part. For example, if we are asking that which part of the ovule is attached to the placenta, then that particular part is the funicles. That particular part is the funicles. Then these funicles comes and attach to an area known as helium. Helium serves the boundary between this inside structure and from the outside, and where the funicles are gone and inverted up, that point is known as the chalacal end or chalacal pole, and we'll be having micropyle. Micro means small, pile means hole, through which the pollen grains come in. And this micropyle, and it is one of the identifying features, how we can identify an ovule. So these are the features they are seeing, where they will see whether they have having micropyle, and they will see whether the chalacal pole or chalacal end is there. Then after that, we are having the outer integuments, inner integuments. These both are for covering, and the integuments will later develop into seed coats, and inside that we are having nucellus, and these nucellus is responsible for megasporogenesis, and this nucellus is having very high cytoplasmic activity, so that it serves as a nourishment for this embryo and growing embryo part. Then this is the main thing you have to know, and this question is asked for the board examination. Either they are asked explain the structure of an ovule and the structure of the embryo sac. We are going to discuss about it, and the next we are going to discuss about it is this is my ovule is otherwise also known as megasporium, and from inside this megasporium one cell will act as mother cell megaspore mother cell, and this megaspore mother cell will undergo meiotic division, and it will form this female gametophyte. So now we are going to study about that, and the next question comes from where this megaspore mother cell comes. This megaspore mother cell originates from this nucellus. This nucellus will undergo division, and that one of the nucellus will become a sporangia, and that particular sporangia is considered as a megaspore mother cells, and this megaspore mother cell will form like this. The diagram is very clearly given here. So this megaspore mother cell is this one. This is one cell, and this is a nucellus will divide into a megaspore mother cell, and this megaspore mother cell undergoes division. That means especially megaspore mother cell is 2n in condition, that is diploid in condition. It will undergo meiotic division, and they form 4 haploid megaspores. 4 haploid megaspores. This is a structure. 4 haploid megaspore. Concept is very simple. Megaspore mother cell MMC, it will undergo meiotic division, and they form 4 megaspores. That means megaspore tetrad will form. And from where this megaspore is coming? This megaspore mother cell is formed from the nucellus, and after the forming of this megaspore tetrad, one among the cell will function as, where is that? One of the cell will function as a megaspore cell, and this megaspore cell will develop into this one, that is this embryo sac. So now we have to study how this embryo sac is formed, because inside this embryo sac only we are having the egg, which is known as the female gametophyte, and this megaspore mother cell is monocarpic in, it is very clearly given here. It is a not monocarpic, monosporic development. Why it is known as monosporic development? Because from this spore only one will develop into this embryo sac, so it is known as monosporic development. If two are there, disporic, trisporic, tetrad, like that. There are some plants are there which will, and from this, after that means it's formed by the meiosis, and from this 4 one cell will further divide mitotically. That means one will divide into 2 will become 4, 4 will become 8. That means 3 mitotic division takes place. And all the cells that is formed will be haploid in nature because it is formed from this particular cell, this particular megaspore, which is haploid in nature. I think it is clear. Then whatever the division takes place, whatever the single cell, when they have divided mitotically, those cells will start to rearrange. When they are rearranging, they will get a structure like this. That means 1, 2, 3 cells will arrange opposite to the micropyle, that means the chelacal end, and those cells are known as antipodals. 2 will arrange to the middle, which is known as the central cell or polar nuclei, which is called as polar nuclei, and egg, then 2 synergids. So when we count this, we can see like that. This is 1 cell, 2, 3, 4, 5, 6, 7. 7 celled, 8 nucleated, or 8 nuclei. That means this is having 1 nucleus, 2 nucleus, 3 nucleus, 3 plus 2 5 plus 3 8. So it will undergo a rearrangement, and they will form a structure like this. And this particular structure, finally what is coming, it is called as the embryo sac, inside which we are having the egg, and this particular process is known as gametophyte. That means female gametophyte formation or megagametogenesis. So this question is also very important for the exam. The question will be very simple. What is the structure? Question may be a direct question. What is the structure of an embryo sac, or what is the structure of an ovule, or otherwise a question will come, how this 7 celled 8 nucleated cells are formed. Any of these questions are possible. So I think the concept is clear to you. Then if you are asking the ploidy means the set of chromosomes inside an organism or a cell. All this thing will be, that means whatever it is formed in this will be N in condition. But this polar nuclei will be central cell, they fuse together and form 2N, that is a separate one that will deal later. So apart from that, all will be N in condition. So you must understand that megaspore mother cell is 2N in condition. Megaspore is N in condition. From that N, 3 mitotic division has happened. They have rearranged, and a structure is